I'd like to introduce our first speaker, who is Shih from the University of North Carolina, Chapel Hill, and he will be talking today about SORDINO. So, I'll hand it over to you.
All right, thank you, Lisa, Lauren, and Sheila for a very kind introduction. I promise to keep this short, and apology for those who had heard the story before, but I'm gonna share with you some a technique now we call SORDINO, which we can use to map brain activity and connectivity changes. So I do have a disclosure to make. I have a US patent for the use of ZTE-based sequence for fMRI, and this covers different trajectory and ramping schemes, including the SORDINO technique that I would talk about. So as many of you know, the gradient-based EPI sequence has been the most widely used approach to map brain-wide functional dynamics. However, every now and then you hear people complaining about fMRI being acoustically too loud. It is spatially unspecific. It's too sensitive to motion, and we all want to have better sensitivity and less distortion. And several years ago, I came across some papers using multi-band Swift technique, and it seems to have the ability to address some of the problems that I listed in the previous slide. But then back then, we did not have access to the multi-band Swift sequence on the Bruker console, which is the MRI system that we have, and also the sensitivity and then the scheme of the way that we sample the data using either the multi-band Swift or also the backbone ZTE technique seems to have some room to improve. So we still decided to give it a try for this ZTE approach, and when I first look at it, it seems to be quite straightforward in terms of forming image. You basically just ramp your gradient to a plateau, and in the condition, you hit an RF pulse. Because your gradient is already on, so this naturally just realize one line of frequency encoding. So you get this free induction in phase signal encoding one line. What you do next is you ramp your gradient to a different directions, you hit another pulse, encode another line. You basically just repeat this process again and again until you fill up the entire K-space, and keep in mind that this is a 3D technique, so you have a non-selective RF pulsing. And there's a little catch here. There's a transmit-receive switch that time, so you lose some data points around the center of the K-space, and that's defined by the hardware. If you look at the sequence in detail, if you grab the ZTE sequence on your preclinical scanner, you'll realize that this is probably not the most efficient way to sample the data, because out of almost a minimal 1 millisecond TR that you can achieve, you have about 30% of the time spending on spoiling the residual signal, and you have 30% of the time spending on ramping and settling the gradient to the next direction. This leaves somewhere around only just 20% of the time that you can sample the data. And as you can imagine, an efficient way to sample the data is actually to spend the minimal amount of time to prepare your contrast and spend the maximum amount of time to sample the data. So we have been brainstorming about how to improve the sensitivity for this so we can actually detect intrinsic T1 related changes. So this was the work led by a grad student back then, Dr. Martin McKinnon, who's now a postdoc in Alan Koretzky's lab at NIH, and also a former postdoc, Dr. Yun-Kong Ma. So we figured that we need to tackle this time-consuming element, and a specific strategy that we came up with is actually very simple. We went through several different iterations, but ended up, we figured that you can actually achieve this three-dimensional encoding with a very, very much simplified approach. You basically just clamp your total gradient amplitude to be constant. So let's say if you have a 20-minute fMRI runs, from beginning to the end, your total gradient amplitude pretty much stays the same. And what we're applying here is a constant rotation angle alpha. So you're rotating the direction that this gradient is pointing to at any given moment. So in essence, your gradient is changing all the time. So let's say, give you an analogy here, if you know having a transient heart rate changes during running the marathon is bad for you, then the potential strategy is that you keep your heart rates almost identical, right? You split the energy throughout. So strategically, when we do this, we can actually reduce the slew rate by several order of magnitudes lower, and in a sense, you can quite comfortably sample the data during the ramping gradient. So this could achieve more than 95% of the sampling time. And you can reduce the bandwidth and improve the signal to noise. And since we sample the data during this constantly changing gradient direction, we will no longer get the straight spoke as you will see in a conventional ZTE, but instead, all the spokes will be curved a little bit in the form that look like this. So if you sample more sparsely, the spoke will basically curve a little more. We use an RF spoiling strategy to tackle the formation of spurious echo, and can also improve the TSNR a little bit. As you can see from the bottom here, these are real life data. And also, one thing that you have seen in many of your MR data is that you don't really have too much of an aliasing problem in your frequency encoding direction. In this type of encoding method, because all the encoding directions are frequency encoding, so we can actually extend the field of view by oversample the data. So since we acquire fMRI data during magnetization steady state, we now call this steady state on-the-ramp detection of induction decay with oversampling, or in short, SORDINO. So this term represents muting of a musical instrument, and we had a chance to call this dinosaur, but we decided not to mess with this. All right, so I'm going to show you some comparisons between SORDINO and against the gold standard grid and code-based EPI, and also the backbone ZTE technique. So on the panel A, we basically fill in the microphones to make sure the sound pressure level, so the acoustic noise profile. You can pretty much see that the SORDINO overlaps with the scanner idle state. And also, this translates to a much reduced stress hormone, cortisol. So each dot here represents measurement from a mouse. So just in a few days of habituation, we can drop the cortisol level to the degree that's pretty much the same as a naive group of subjects that never experienced MR scans. And in our hands, on our particular setting, it seems hard to achieve so with the EPI protocol. So because we can reduce the slew rate by a lot, this gives you the ability to accommodate some of the multi-modal recording. In these two examples on the left, these are electrophysiological recording with a very well-known established ground truth. On the right, these are electrochemical recordings. So you can kind of see the improvements not only against the gradient echo-based EPI, but also improvements against the backbone ZTE technique. So in essence, you can put in electrodes, do your E-phase recording, and even just use the online sorting tools that's available from your electrophysiology software vendor or hardware vendor. You can get to single spike precision. So the method is pretty much resistant to ghosting artifact, and it also doesn't have distortion problem as you commonly see in EPI. And more importantly, since this method essentially has no echo time, so you don't really suffer from the susceptibility artifact that you commonly see in the rodent brain around the amygdala area, or for example, in humans, you might see that in the frontal cortical region. So the method itself is also quite resistant to motion artifacts. So typically, if you can fix the head quite well, you can really limit the head motion to a degree. But another factor coming from the common motion artifact is really motion from outside of the head. For example, you kick the leg, you're breathing, and you're moving the arm, things like that. So these elements are actually outside of your imaging field of view, but actually they would distort the homogeneous magnetic field and cause motion artifact and distortion. So what we're doing here is that we're tying a fishing string to the forelimb of a mouse, and we pull it by sitting outside the magnet. By doing so, and this is a deeply anesthetized mouse brain. So when we look at the activity changes in the brain, we can run the correlations against this motion paradigm, and in the EPI scans, we see several voxels showing very strong positive and negative correlations against this motion paradigm, but in SORDINO it's pretty much centered around 0. And you can also benchmark the motion parameters and also look at the frame-wise displacement and see some improvement there. And perhaps one of the important findings that we have from this recording scheme is that if you select your TR and flip angle properly, in a within subject comparison, so these are within subject and within session, same stimulation, same subject, and a matching spatial temporal resolution, we can show that SORDINO is not carrying as an inferior signal to noise as compared to, or contrast to noise as compared to the gradient echo EPI scan in this 4-pole task. And you can also use this to map functional connectivity. So I don't have a lot of time to talk about mechanism today, but we believe that this sequence, when we measure without a lengthy echo time, it captures tissue oxygen changes and also capture some of the perfusion elements that's originated from the arterial CVV changes that brings in stronger magnetization. We did some phantom study to look at the inflow effect. So when you have a fresh coming in spins, they actually carry stronger magnetization. So as they will receive less number of RF pulses, they're not reaching to a saturated steady state. So they carry a stronger signal. So we know that when you use a head-only transceiver coil, this will actually give you some of the inflow elements. And this can actually be seen by doing an experiment by comparing a fMRI response from a single subject, but then use two coil excitation schemes, head-only excitation coil versus whole-body excitation coils. And indeed, as we modeled, and the detail can be seen in this bioarchive preprint that we put up a couple months ago, with the head-only excitation coil, you indeed had this extra contrast boost as compared to using a whole-body excitation coil. So we know we're not measuring the bulk contrast because we did some validation studies by drawing the blood from arteries and the veins. And you can put this freshly drawn blood into a phantoms and scan it with conventional T2-weighted protocol, and you'll see the typical bold effect, as you see on the right side. But if you scan with the SORDINO sequence that we use for actual fMRI scans, the venous blood actually carries a slightly stronger signal as compared to the arterial blood. So at least the signal is not coming, it was not having a strong weighted from the intravascular oxygenation effect. However, through some of the benchmarking experiments that we use, fiber photometry to measure the non-physiological ground truths, and also using an oxygen electrode to look at tissue oxygen changes, we performed an experiment that we dissociate the blood volume and oxygenation by using a gas mixture, this kind of gas challenge. And in this case, these are concurrent lead measures, and we're picking the SORDINO signal just from a single voxel at the tip of the recording fiber. We can kind of see that we can dissociate the CBV and increase the blood volume while decreasing the oxygenation. And SORDINO signal actually tracks the oxygen, actually follow the oxygen changes quite well. And in a separate experiment that we use a different ground truths oxygen electrode measurement, the SORDINO signal also follows the FSCV-derived tissue oxygen changes quite well in both this case and also the hypoxic challenge. So this suggests that the contrast actually also have a strong tissue oxygen origin. And if we are indeed measuring tissue oxygen and very localized arterial CBV changes, this means that this method could be quite spatially specific. So just I want to show you some application studies here. So in a common experimental scheme that we're running right now in the lab, basically use this type of head fixation plates that also couple directly with the coil. So the coil traces are directly on the fixation plate, so you can actually minimize the distance between the coil and the brain. So the animals are wearing these plates, and we can image them for a long period of time. So you can then process your data by doing either simple C-based analysis to look at connectivity changes or pull ICA derived brain features. You can also port your data to the standard Alabrain atlas. So on the upper right, this was the work by my colleague Sun-Ho Lee here. On the upper right, these were structural connectivity ground truths that you pull from Alabrain. You can use the same ROI to pull functional connectivity from our SORDINO scans in awake animals. And I think we're also taking one step further toward mapping brain activity during active behavior. So this shows you a mouse being trained for this foot palate grabbing and eating task. So the animals are waiting for this palate to come, and as soon as the pallets arrive, they grab it very rapidly and chew it. So we can actually do this inside the scanner. We add a little mirrors on the side to better visualize the process, so this will facilitate the later video analysis. But this panel D here shows that we're dropping the pallet at the timing that, this red arrows pointing down. We can drop this pallet repeatedly, and when you draw an RI in the contralateral motor cortex, you can consistently see the activity changes. But if you also look at the brain-wide activity changes here, at the first time point the pallets arrive, we see the suppression of the default mode-related area, and then following that time point, you see bilateral thalamic activity changes, and also the cortical signals start to engage, and also some changes in cerebellum. So we also developed a scheme to image two animals simultaneously. So there's a separator plate that separates two animal subjects here, and you can see that there are two animals, each wearing a head plate coil inside the scanner. So with this scheme, we can actually image two brains simultaneously. So instead of just looking at the connectivity or activity changes within a single subject's brain, with this scheme, we can actually look at the coupling between the two brains, or look at different brain regions, how they synchronize and desynchronize, and just expand the capability for us to address a variety of different type of neuroscience questions. And because we don't have a lot of penalty coming from susceptibility artifact or electromagnetic interference, you can now quite comfortably bring a calcium miniscope inside your MRI scanner. So this is an example that we bring, that we look at the calcium neuronal activity in the prefrontal cortex of a mouse brain, and this is a nearly real-time image here. We can get somewhere around 100 cells in every single session, and we have been imaging these animals for several months. And this allows you to address some questions like, whether or not they are distinct cluster of the cells, they are associating with different large-scale brain networks in the rodent brain. And you can also expand this type of technique to address different type of neuroscience questions. You can also do things beyond fMRI, since this method is capturing T1 weighted signal changes. And this example was the injection of gadolinium through the cisterna magna, so we can look at the glymphatic clearance. But again, the benefit of this is to improve quality of the images and reduce the susceptibility confound. And also, you can do this type of imaging in the wake or in the sleep conditions without anesthesia influence. You can also capture manganese-enhanced signal changes. The video on the left shows you, the image before the baseline, before the injection of manganese, and then on the right is 24 hours after IP injection of manganese. So you can kind of see that the manganese is accumulated in the brain and through a calcium-weighted pattern. You can also do acute injection of manganese to pick up the signal changes within the ventricle, specifically in the coral plexus area. And these traces are actual data. These are not coming from the modeled results. So you can kind of see a much improved quality here. And we can actually assess the kinetics and see how this manganese are being transported through the paraventricular space. All right. So I walk you through a method called SORDINO. And it won't replace gradient and echo-based EPI, but it has several features listed here that's worthwhile for animal fMRI people to consider. And all in all, I think if you use the techniques that they can use short acquisition delay methods, and as long as you employ comparatively less aggressive gradient modulations like the one that I introduced to you, this actually holds potential to further enhance the benefits that we see in the list here, and this will facilitate animal fMRI in a wake behaving condition. And people with their name highlighted here really did the work, and I thank you for your attention, and happy to take any questions.
I always learn something new each time I mean, I've only seen it once or twice, but I learned something new. So you talked about, the sensitivity to tissue oxygenation, and that would potentially make it, more specific. But, T1, due to oxygenation, changes in big blood vessels too.
Correct.
And of course, you also kind of emphasize that there is a component of inflow, which of course occurs in big vessels. So is that really any different than the problems we have with BOLD? That side it's only the vein, but now you have the artery and the vein that could be disturbing the localization in some way.
Right, so you can actually use different type of RF excitation schemes to dissect this further, right? So in a brief experiment, in an image that I showed, when you use a volume coil for excitation, basically your blood signal would approximate to a lower steady state, and because blood has slightly longer T1, right? So the steady state level will be slightly lower than that of the tissue of the gray matter. So basically, from our observations, by doing that experiment in a single subject and we just switch the excitation schemes, that seems to suppress at least 50 or 60% of the evoked response in our hands. I do believe that the inflow element is a little different from a single slice macroscopic through plane inflow, right? Because in this scheme, we are doing non-selective RF excitation, even using a head transceiver. Right, there's non-selective RF excitation to the entire coil B1 field. Of course, there are flip angle differences as you go further, but essentially, the incoming blood will continue to receive additional number of RF pulsings as time goes by. So when you go to the venous side of things, I believe the mechanization will be much lower. So we'll actually bring in much stronger arterial input signal as compared to the venous signal.
I was also wondering whether maybe you get some kind of gradient spoiling effect for the flow because of your sequence, but I'm not sure with radial-like sequences whether that's true or not.
Yeah, well, that probably requires further validations to say for sure, yeah. Thank you for the questions.
Other questions? I think we're fine for sake of time to.
All right,
stick to
thank
the
you. Well,
slide,
I'm
but-
around, so happy to discuss further. Thank you.
Thank you, Ian.